Hi, I'm Harper and welcome to Harper Carroll AI. If you don't know me, I'm an AI engineer, advisor, and educator with about 10 years of experience engineering AI and machine learning at Stanford, Meta, and then I was founding engineer and then head of AI, ML at a startup acquired by NVIDIA. My goal here at HCAI is to educate everyone about AI so that you can understand it and use it to your benefit. The following is a conversation with Noor Siddiqui, founder and CEO of Orchid Health, a San Francisco-based biotech company transforming reproductive technology. Noor is a Stanford-trained computer scientist and former Thiel Fellow, and she was inspired to launch the company due to a personal experience. Her mother's diagnosis with retinitis pigmentosa, a genetic disease that causes progressive vision loss, despite the fact that they had no family history. This caused her to launch the company in 2019 with a mission to help people have healthier babies with full genome embryo screening. Basically, what the company does is it allows couples undergoing IVF to test for various genetic conditions, over 1,200, like cancer risk, neurodevelopmental disorders, and heart disease. The company also offers genetic screening for adults at a lower cost. Since its debut, the company has raised from investors like Prometheus Fund and Figma CEOs Dylan Field, and its technology is now available at IVF centers nationwide as of 2025. It has caused a lot of excitement and also controversy in the world. There was actually just a New York Times article written about Noor and about Orchid, and there's some questions about, the ethics of this all. But this isn't about, choosing your child's eye color. This is about finding very serious conditions, often life-inhibiting, that you can prevent. If you're already doing this kind of screening with your embryos, then you may as well get more results and you can pick and choose what you get. But I would listen to this, see what you think, and let us know in the comments. But Noor is a very special person. She has an infectious energy. I know that she's doing this for all the right reasons, and I'm just so excited to have her on the show. So I hope you enjoy our conversation. Noor, thanks for being here.
Thank you so much for having me. This is badass. You're badass.
You're badass.
Oh, thank you.
Okay, let's just jump into Orchid because you have a pretty amazing company and I want to hear all about it. You're a badass. So let's talk about it. Just tell me the spiel on Orchid.
Yeah. Okay, the spiel. Well, the main spiel is that we want to help everyone have a super baby.
Ooh, okay. Okay. Full genome embryo testing.
Yeah, exactly. So kind of like the backing up the story for how this all got started is that I was at Stanford like you and, bright-eyed and bushy-tailed. And the thing that was really most exciting to me, I was working in a bunch of different AI labs and I got kind of really fortuitously scooped up into a genomics and AI lab. And I was like, whoa, this is crazy. All that data, millions of people's genetic sequences are now actually here, right? Back in 2000 when the human genome was first sequenced, there was this promise of one day we were going to decode it and we were actually going to be able to have impacts on our health. But for me, the thing that was most interesting was I watched my mom go blind from a really young age, and the condition that she has is called retinitis pigmentosa, and it was this odyssey of meeting a ton of different specialists to actually get that diagnosis. And I was kind of on the cusp of getting engaged at the time, and babies were not this very abstract concept anymore. It was sort of very concretely, with this person, right? Yeah. And the biggest thing on my mind was that, I don't want my child to go blind, and I don't want them to be affected by, unfortunately, the thousands of different genetic diseases that are available. So we kind of just got cracking with some of the best single cell sequencing folks at Stanford, some of the top embryologists and fertility experts in the world. And we kind of had this tag team of people come together, work really hard on R&D for a couple of years. And now we're the first company in the world to be able to do whole genome sequencing on embryos.
Oh my gosh. Okay. So there's a lot here.
Yeah.
I want to back up so that the listeners know what we're talking about. So in case you don't know, full genome embryonic testing is when you have an embryo made via IVF.
Yeah.
You retrieve the eggs from the body via egg retrieval.
Yeah.
In like an IVF clinic, right? And then you make an embryo by adding the sperm, and then you choose the embryos, and then you're now able to run tests on these embryos. They already do standard testing for embryos first, right? Before they decide which ones to use. But now you've expanded that testing sweet.
Yeah, yeah, exactly.
Enormously.
Yeah. So kind of backing up how does IVF work and how does this whole process work? So the female partner is on medication for about, anywhere from 7 to kind of 12 days. And what she's doing is she's maturing the follicles in her ovaries. So your follicles on your head produce hair, follicles in your ovaries produce eggs. So you're on that medication for about, 10-ish days. And then your eggs are retrieved. And then there's actually one of the things that's really fun to do at Stanford that kind of made me fall in love with, honestly, the whole process is you have these eggs that are in a vial and then they get dumped into a petri dish. And there's an embryologist there literally fishing the eggs out of a petri dish. And the speed at which she does that fishing expedition for your eggs determines your entire funnel of how many embryos you're going to create, right? Because those eggs are so precious. And those eggs are then fertilized with your partner's sperm. They're grown for 5 days. That's when they move from an embryo, which is kind of that single cell stage, to become a blastocyst at day 5. And then there are about 125 cells, and that's kind of where Orchid comes in. So there's been a standard biopsy or cell sampling procedure that's been going on for over 15 years to actually get samples of cells from the trifectoderm, so the outer edge of an embryo.
Yeah.
And we take that exact same sample, so those 5 cells, and we're just able to generate 100 times the data. So that whole genome that you can get, on blood or saliva, that's easy. But on embryos, you have about 30 picograms of DNA. So you have 10,000 times less DNA than you need. That's kind of where that sort of Orchid magic protocol comes in to be able to amplify or copy that DNA, over 10,000 times so that you can get that whole genome off of that really, really tiny amount of DNA.
Oh my gosh. Okay. There's so much to go into here. But one thing that I think is really cool about egg freezing and egg retrieval that some people may not know is that when you retrieve eggs, you would be losing them anyway. Like every month your body makes a bunch of follicles in the ovaries and then one is chosen to become the egg. Otherwise, the rest of them just leave your body with your menses. But basically what the medication does is it causes as many follicles as possible to turn into eggs, right? And then you just retrieve them as opposed to them just being dumped. So that's just a cool thing. It doesn't really have to do, but I think
that's a really good point because a lot of people think they're kind of nervous about doing an egg retrieval because they're like, oh, I'm going to accelerate menopause. But that's a huge misconception. Yeah, it's not, you're not stealing eggs from the future. Unfortunately, you're totally right. You lose eggs every month and
use it or lose it.
Exactly. So yeah, it kind of gets you.
Okay, so let's back up. So you were at Stanford, you did your undergrad and master's in computer science like me.
Yes, twins.
and then you were working at a genome lab. What was it? You were at a genome, you were doing cool genome research.
Yeah.
And, tell me about that.
Yeah, so the PI in the lab, his name is Anshul Kundaje.
PI is a
principal investigator. So I was rotating in a bunch of different labs. One of the first labs I was in was Sebastian Thrun, who's like the most badass person in my opinion. Like he, started Google X, and he's kind of like the guy behind the self-driving car. So yeah, we're working on like melanoma versus carcinoma, so skin cancer classification first. So I was kind of like,
and that's AI for AI people classification, right?
Yeah, exactly. It's kind of funny because that was like the cover. It was a cover Nature paper at the time, but nowadays it's so chill. People are like, that's, you know what I mean? It's kind of like table stakes. It's like it's insane how much progress AI has made in just a small number of years. Because that was state of the art. Not even.
Congrats on being on the cover of Nature. I hear that's a
no. It was a lab. It was a lab. That was before my time.
Okay,
but it was like it was amazing to be a part of that lab and see what they were able to build. But yeah, so then when I came to Anshul's lab, he was interested in something called transcription factor binding. So transcription factors are just certain segments of DNA that are kind of interesting from a biological perspective.
Okay.
So it's kind of more of a basic science problem that he was interested in, but through his lab, I was able to kind of get exposure to just building models on instead of image data, but genetic data. And the thing that I think is really powerful is that previously, there wasn't a way to really quantify genetic risk. Yeah. And now, because these data sets exist, you can quantify genetic risk for things that previously all you could do is give an empirical estimate. So for example, like let's say you have a sibling with breast cancer or schizophrenia or bipolar disorder, right? You can kind of empirically estimate that, okay, those families are at higher risk, but you can't actually quantify for an individual what their risk is before these models existed. And now that these models exist, it's super, super powerful in the IVF and embryo setting because, if you have a familiar risk for, schizophrenia, bipolar, there's not a lot of ways to mitigate risk. And being able to, at the embryo stage, select an embryo that has fewer genetic risk variants. You can basically, go from, the 99th percentile of risk perhaps for one of your embryos to the 60th or 70th, which has like a, 40 to 60% relative risk reduction. It's a lot of numbers, but basically, the way that you can think about genetics is that you kind of have marbles in a jar, right? And those marbles are kind of getting randomly assorted in embryos. So sometimes you kind of get unlucky and you get a lot of marbles in a jar, which you can kind of think of as a lot of risk variance. And other embryos kind of get lucky. You can kind of think of it as like, hey, there's like a genetic lottery that's happening.
Right.
And instead of that information being, you being blind to that information, you're actually able to see that, not even as an adult or not even as a child, but pre-pregnancy.
So this data set that you're talking about that you were exposed to, I think our listeners like to learn about like AI and what these data sets look like. And like as AI advances and we're able to gather more data, we're going to get these enormous insights, right? So the data set that's been developed in the genomics area that you're in, what is that? Is it basically just like DNA sequence mapped to different like diseases that people have, or like how does the data set look?
Yeah, exactly. So you basically, what is a DNA sequence? It's like a sequence of letters, right?
Letters,
A, T, C, G. And the way that I like to talk about genetic mutations is they're literally just like typos, right? So the same way that you do spell check on, a document, what Orchid is kind of doing is spell check on the genome, right? So previous testing, it's called PGTA or chromosomal testing. Okay. So what that's looking like is you're kind of doing spell check on only the table of contents. So how many chapters are in your book, how many chromosomes are in that embryo's genome. So if you have an extra or missing chapter, that's unfortunately not compatible with life. But that's really missing a lot of the detailed information, right? Like you wouldn't want to only spell check the table of contents. You'd want to spell check the entire book. So any way that you can mess up a string or a word is the same way that you can unfortunately create a genetic anomaly. So
really? So it's basically like tokenized.
Yeah.
Wow. So the genome is represented as a string or just like a series of letters, 4 letters, A, T, C, G, I think it is.
And you have 3 billion of them to make one genome. 3
billion of them. And you're able to, as you said, kind of like scan through and you know what normal sequences look like. Are they all like the same length? So you would say like every 4, you're checking that it looks like a normal one, or like
so like basically genes, you can think of them as like subsets or like certain strings. So genes can be anywhere from thousands to tens of thousands of bases long. And the way that you define a gene is there's something called a start codon and a stop codon.
That's how you do it. Okay, cool. Yeah.
But
kind of like a start token and then token.
You can think of it as like a capital letter and a period, right? And you can think of genes like sentences, right?
So cool.
Yeah. So basically, it like think about anything that can go wrong in a sentence or like literally a typo, right? You can have a insertion, right? An extra letter, like instead of a cat, it could become coat. Those are really cat and coat, mean really different things.
Yes.
So, dog and dig mean very different things. So you can think of it like that. There literally are something called insertions, deletion. So an extra letter, deletion, a missing letter. There's frame shift mutations. It sounds like you talked about that, where if you add a letter, then, unfortunately the protein that's produced changes depending on where that letter is inserted or deleted. There's a lot of different variants that can happen because of these typos. And what's really cool that's happened over the last 2 decades is that geneticists have cataloged thousands of different mutations and what disease they actually map to. So that's kind of
so cool. So I guess that's okay. So you're scanning this sequence of genome tokens or genome letters. Okay. Yeah. And you're able to, is it context dependent? So you were saying like dog and dig, are they noticing like, let me back up. So they're scanning this genome sequence and then they're looking, they find the start, they find the stop, and they're saying, does this word or sentence, I guess is the stop at the end of the word or the sentence? I guess it's kind of like a molecule, so it's like multiple. You can't think of it perfectly similar.
Yeah,
but
it's not exactly the same, but basically like
they see what's normal.
So some sentences are short and some sentences are long, right? So some genes have thousands of letters, some have tens of thousands of letters, right? So it's the same as a sentence, right? But there's basically a way that
I see. So you're scanning and just making sure that there is some map to a normal genetic sequence that you're looking at right now. Is this segment mapped to a normal one? And if not, like what is different about it?
Yeah, exactly.
And then what you see based on what's different, you find the disease. And that's what you're doing.
Yeah, exactly. So you can think of diseases as like typos, right? So basically chromosomes are like chapters, right? So they're really, really big structures, right? So each chromosomes are actually laid out from smallest to largest, right? So chromosome 1 is like your largest, and then your sex chromosomes like X and Y are actually the smallest. And I think just fun fact for female genetic superiority is that the X chromosome is much larger than the Y chromosome. We have much more data. So
Really?
XX, there's actually this really funny theory. I got to send you the link to this, but basically,
So I mean, more complex. Anyway,
yeah, but basically one of the theories for why women live longer than men is that actually because of our genetic superiority. So, so what that means is that we have two X chromosomes, right? So we have basically two copies of DNA versus for men, unfortunately, they have just one, right? XY is male. Okay. So basically our cells actually have kind of more redundancy is kind of the way that you could think about it.
So you're saying we have two copies of DNA. You're saying because everything on the Y is different than on the X?
Yeah. So we have two X chromosomes and men have a Y chromosome. So Y chromosome has way less data than the X chromosome.
Okay.
X chromosome is way bigger.
Okay.
So,
I see. Yeah.
Yeah. So there's something called X inactivation that happens in women. So basically some of our cells have DNA from one of our X chromosomes and the other ones have DNA from the other X chromosome. So I don't know if you've seen something called like a calico cat, but basically these cats are spotted, right? So that's kind of like a visual way to express it of, okay, like some, fur is, white and the other is, brown or red or all these different colors. So the reason why cats have that mosaic, it's called a mosaic pattern, is because they have kind of different DNA that's expressing in their skin cells. So you can think of females as kind of, where all of our cells are mosaic or kind of spotted.
Because there's like one option for
yes.
There's two X's, and so it can be something on one and something on the other.
Yes, exactly. Right. Versus men, genetically less data, they only have one.
And that one option on the X, that's it.
Yeah, and there's also a whole set of disorders called X-linked disorders where, unfortunately, males are more fragile because they only have one X versus we have two X's. So, sorry, I'm really into this.
Right, because if there's one mistake on their one X chromosome, then that's it. They don't have the chance to make it up on the other one. Yes. And that's why calico cats are always female.
Yes. So anyways, basically
this is cool.
Yeah, just basically backing up on this whole chapter thing, right? So chapters, right? So you have your chromosome 1, which is your largest, and then your sex chromosomes are your smallest. Yes. So basically, like Down syndrome, for example, is actually trisomy 21. So you have 3 chromosome 21s, right? So that's actually a really rare situation where you have an abnormal number of chromosomes and it actually is compatible with life. It is a disease, but it's compatible with life, right? Gotcha. So if you're missing a chromosome 1, I don't think there's ever been a reported birth of a baby born who is missing a chromosome 1, right? Because there's just so much data on that one chromosome. So chromosome 1 has, there's 20,000 genes total. I don't know how many exactly are in chromosome 1, but it's one of the largest. So probably has like on the order of like, 1,000 plus genes. So you can't be like a human without chromosome 1.
So chromosome 1 is very long as well.
These are much bigger.
It has like the string of
letters
is very long. Okay, because it's holding all.
But no, you're right. The way that proteins work is it is kind of tokenization, right? So basically, those letters are broken up, and then those create amino acids or kind of building blocks to proteins. But yeah, it's really crazy. There's kind of like a tokenization process running in every single cell in your body, at the same time to keep you alive.
So if you are not familiar with tokenization yet, it's like fragments in large language models. Mm-hmm. You have fragments of words. I know Noor already knows this, but you have fragments of words, and basically the model is outputting token after token after token. So fragment of word after fragment of word after fragment of word. And so in the case of genomics, the fragments of words, of molecules would be fragments of proteins would be amino acids.
Yes.
So you have these tokens that are these pieces of the proteins that come together to make sentences, which are proteins, like in a line, which is your genome.
Exactly. And the other fun thing about amino acids in DNA is that there's actually some redundancy in basically amino acids. So basically different sequences of letters will make the same amino acid because those amino acids are kind of like more important. It's kind of like just, I don't know, it's kind of like some magic of biology in some ways, because basically when DNA is copied, it doesn't get copied perfectly, right? Unfortunately, there's like slippage, there's errors that are introduced naturally, right? And I guess the reason, I don't think anyone really knows about how this was designed, but the theory is that there is some redundancy built into the system because, once every, 10,000 or a million bases, there will be an error introduced. There is also an error correction capability that DNA has too. Isn't that crazy? Like everything that we, kind of put into like text interfaces, like in our Google Docs and stuff, are also in your DNA. But yeah, there is kind of a built-in enzymes that are spell checking and kind of correcting errors. There's a template, right? So you have two pieces of DNA that are, A's bind with T and G's bind with C's. So there actually is a kind of templating and like error correcting process that's happening.
Natural intelligence. Like we have our spell check artificial intelligence, and that's the OG intelligence, right?
Yeah.
Yeah.
No, yeah, totally. And anyways, I just think it's interesting that through that process, there's still some redundancy built in. So even though these some spelling errors are introduced, because there's redundancy in how amino acids are formed, even some spelling errors are innocuous. But unfortunately, there's thousands of spelling errors that are not innocuous, right? Like if you're in a single letter change in a critical spot can be the difference between life and death, not only like in the womb, but after that, like in infancy, right? Like, some studies have found that, up to 40% of babies in the NICU are there for a genetic reason.
Wow, 40%.
Yeah, really, really high.
Wow. And so you've been able to find out which of these spell errors are incompatible with life based on this AI data set. And that's what you use in order to help find the embryos that are the healthiest and have the fewest diseases, I guess.
Yeah.
Right?
So I would say the big places where AI is used is in the models for complex disease and on variant interpretation. So when we're talking about kind of the monogenic stuff, that's actually the really hard work of geneticists over the last 2 decades, right? So as more and more people got sequenced, they cataloged these thousands of diseases. So there's actually 30 million Americans, so about 10% of the US population that has one of these rare genetic diseases. So they're individually rare, meaning a rare disease is defined in the US as under 200,000 people have it. But if you add up all the thousands of rare diseases, it actually ends up being a really large fraction. So that fraction you actually don't need AI for. That's something that has kind of been cataloged,
It's just matching.
Grueling work. Yeah, it's kind of like
matching
over, took 2 decades.
Yeah.
Where AI comes in is for these more complex diseases. So for things like schizophrenia or heart disease or bipolar, where it's not just one typo causes one specific disease, but where it's the cumulative impact. It's kind of like you can think of it as DNA paper cuts almost, right? It's like every time an enzyme or a protein isn't working quite perfectly, you kind of have a diffuse negative impact on your health. It's kind of how.
Oh, that's interesting. So that's how it is. And so for some of these illnesses, as you said, like schizophrenia, you actually need these minor errors, spelling errors in many places that over time just kind of overwhelms the body or genome of the person.
Yeah. So I wouldn't call them errors in the same sense as so with the monogenic diseases, I would agree with you. It is kind of like spelling errors. For the polygenic conditions, it's more like spelling differences, because each of those differences, they don't cause a disease. It's kind of like there's a diffuse, it's like paper cuts.
Yes,
it's like you're not
it's not an error itself. It's just, and the way that you've been able to do that is with AI, seeing like, okay, this person's genome maps to schizophrenia. And then as you have enough amounts of these people's genomes, you're able to see, whoa, like we've noticed that in these 10,000 people with schizophrenia, they all have this one little spelling variation.
Yes.
And that's where the AI comes in.
Exactly.
Because it's able to process massive amounts of data. And see
and
what features are
exactly.
similar.
So we would never be able to look at this by hand, right? It required hundreds of thousands of individuals, where, kind of going back to what the data set looks like, you have sequence data, so those A, T, C's, and G's, and then you have that mapped to their medical record. So their medical record can include all sorts of information. It can include what drugs they took. It can include psychiatric evaluations. It can include their own self-reported data about their symptoms. So there's nurse-verified, physician.
I see.
So some data sets are richer than others, but obviously, the best ones are richer. They have more. They have, LDL levels, HDL. They could actually have lab results inside their report. So you can kind of define your model arbitrarily depending on, what is most important. So like if you have a certain physician who really wants to define heart disease really narrowly, they might say, I don't want people who are just on statins or on a specific medication. I want them who've actually had a cardiac event, right? You can make your model more or less stringent depending on how you define heart disease. How do you define schizophrenia, right? Which is really cool, because now you can actually really get to kind of a closer to a molecular understanding of these diseases because, certain diseases are really vaguely defined, right? You can think of it as like, so for something like height, there's not really a way to fake it, right? You kind of get up to the wall and, how tall are you? Versus for something like, depression, it's really, really wide variety of symptoms, right? Some people are, have made a suicide attempt, right? That might be, more extreme versus other people, may just be struggling with, motivation and
Right.
it's much milder. So it's
Right.
really hard to build a model if you don't define your phenotype really tightly.
I see.
So that's one of the things that's super important is like really working with a, specialist who kind of knows kind of a lot of those clinical features that are going to be really important to get from your data set. And because there's actually a lot of misdiagnosis in medicine, right? So if you get that misdiagnosis to tarnish your data set and how you're defining the disease, that can affect your performance.
Yeah. So in other words, we have this data set, right, of people's features, diseases that they have, symptoms that they're experiencing, whether it's lack of motivation or whether it's suicide attempt, right? So it's people have these profiles and then it's mapped to their genome. And if you're trying to find correlations between genome and symptoms, you want to basically make sure that your definition of what symptom or disease the person has, I guess disease is kind of binary, right? Like you either have the disease or you don't. But in terms of symptoms, you're saying like for depression, if you're trying to find a correlation between genome sequence and depression, you're going to probably want to make sure that you have really extreme cases of depression, like suicide attempts, to find that correlation the best, I guess. Or I guess you could broaden it, but then, right, you have this issue.
If you lump a lot of different things together, then it tends. There are some models that perform better with ensembling, but our users typically have like come in with a very specific thing, right?
Yes.
They're really interested in schizophrenia, or they're really interested in bipolar disorder, or they have a family, their partner has type 1 diabetes, or there's like something specific that they're looking for. So that's kind of why we build these models with the end use in mind, right? So kind of like the way that the output ends up looking is that you find variants that are enriched or, more common in people with the disease as opposed to healthy controls, right? And then in terms of, how those are validated, you look at thousands of people who are in a test set, and I'm sure you guys have talked about trained and test sets.
Yes.
So in that test set, the model hasn't looked at those samples, and then you see, okay, how much more often do these individuals get the disease, right? So maybe you'd get a genetic risk score in the 99th percentile, and then you would be 4 or 5 times the base rate of the disease. So maybe let's say something like breast cancer, let's say, you have like a 10% population average risk. So in the 50th percentile, the average person has a 10% lifetime risk. Then if you're in the 99th percentile, if you're at 4x risk, maybe you'd have about a 40% lifetime risk. Does that make sense?
I guess I just want to back up. So for the people who are new here or do not remember training versus test sets.
Yeah.
So training sets are when you have the labels of the data. So in this case, it would be the genome, or would it be the genome mapped to the symptoms, or what does the data set look like?
Oh, the training set is just the individuals that you're building the model with. So the genetic data that you're using to determine.
The genetic data mapped to like what's the label, like their illness,
Yeah.
or I guess you have individual models per illness. So it would be like, does have schizophrenia, genome sequence, yes, no. Like so it's individual models per illness.
Yeah, yes. And then for each disease, there's a lot of different basically what's called like ICD-9 codes that go into defining that disease. So basically, there's not. So for example, for schizophrenia or for heart disease, it might be, okay, you're on this specific medication, or you've had a heart attack event, or a psychiatrist has diagnosed you.
I see.
So it's not just kind of one label. There's dozens of different inclusion or exclusion criteria for did you meet the definition for this disease, if that makes sense.
Yes, exactly. So it's not just like a binary yes, no. It's like you could exist anywhere on this spectrum or have these different features of someone with schizophrenia. And that's the Y label. That's the classification of the person.
Yes, exactly.
Okay, so the data set is what you use to train the model. So you have individual models for each disease where you have the genome, and then you have this like, what features of the disease are they showing? What medications are they on? What you were just saying.
And then you use that to discover variants that are enriched in the people with the disease as compared to the healthy controls. And then that's how you train the model. And then when you test the model, you basically take new individuals who you haven't trained on.
Yes.
And then,
So sorry, when you say variants, just want to make sure that's clear to everyone. You're saying that the genetic, you're able to find by training this model, you're able to find which genomes then correlate with having schizophrenia or different
So
features.
it's not the entire genome, it's just certain letters, right? So there's certain letter differences
Okay.
that you find that are enriched in the people with schizophrenia as compared to the controls.
I see the variances, the slight
Yes.
differences.
Yes, the
in
letter
letters.
differences,
You
yes.
find exactly what those letter differences are interesting or
Yes.
where they are in the
Yes.
thing. And then you have the test set where you have, given the genome, how likely are they to have schizophrenia?
So there's one more step. So basically,
Yes,
when you're training, so you find those variants that are enriched in the people with the disease as compared to the healthy controls.
Yep.
Then you run those. You basically create a genetic risk score. So basically, you're able to basically compute a score for every single person in that set, and then you create a normal distribution. So some individuals are going to be, you know, in the 50th percentile, some are going to be the 25th, some are going to be the 99th, right?
Yeah.
And then you see, okay, what is the odds ratio or what is the likelihood that someone is going to get the disease compared to the baseline risk, if that makes sense?
Yes.
Yeah, so basically you calculate an odds ratio, you calculate, basically for, it's a little bit technical, but it's odds ratio per standard deviation, right? So when you have a normal distribution,
Yes.
how much more likely per standard deviation that you go out from normal.
Yes, I see.
Basically, if you go 1 standard deviation, 2 standard deviation, 3 standard deviations, how much increased risk are you adding? So basically, models that perform well have more stratification. Models that perform poorly have less stratification.
Stratification in terms of like the standard deviations.
Odds ratio per standard deviation.
Right.
Yeah.
I see. And so you're calculating like a likelihood of having schizophrenia or whatever disease it is, rather than just like, will you have it or not? Right. You have a likelihood, and then you're kind of plotting on this, you said it's a normal distribution. And then the standard deviations are like differences from like 50% likelihood, right? I guess, or maybe it would be at 0.
Yeah.
Okay.
Yeah.
And then, so as you get further and further outside the mean, the greater standard deviation, you are much more likely to have this disease.
Yeah, exactly. So basically what the model is learning is which variants are correlated with an increased risk. So once you have those variants, then you're plotting all those individuals on a normal distribution. And then what you're doing with the test set is then you're saying, for new individuals who weren't in training, how good is my model? Meaning, so for people who are in that 99th percentile, how much more likely are they than the average person to develop the disease? And if you perform well, that means, you know, what performing well means is that it's compatible with your training set, right? So basically, if your training set gave you like a 4x or 5x increased risk of the 99th percentile, you would want to see that duplicated in your test set.
In the test set.
If it fell apart, if it's like, oh, you know, now my 99th percentile is the same risk, then you're like, oh, clearly something went wrong here.
I see. Okay.
Does that make sense?
Yeah. Okay. Yeah, this is technical stuff.
Yeah. Maybe.
I almost feel like we're going too down, but it's I love it's so
Yeah.
interesting. But
yeah, I mean, at the high level in terms of like why this is like interesting or useful to parents is that previously there wasn't a way to quantify your specific genetic risk, right? So for example, South Asians, we get heart attacks, you know, 10 years earlier, and we're unfortunately twice as likely to die as, you know, Caucasians. It's just, you know,
I didn't know that.
Bad genetic lottery, bad genes. And I don't want my children to suffer from that. I don't want them to just be condemned to like having, much higher risk for heart disease. So when, me and my husband ran our embryos, we can now see what is each individual embryo's heart disease risk. So are they in the 99th percentile? Are they in the 70th percentile? What is their absolute risk of the disease? Meaning, so do they have a 20% lifetime risk or, 70% lifetime risk? So for every single disease that matters to you, you can now see before you're even pregnant, what is your embryo's risk of that disease? Yeah.
That's amazing. Amazing. And I mean, many people are doing these tests anyway when they're considering implanting an embryo. So you may as well get all this data instead of just the data that you had before. You know what I'm saying?
Yeah, exactly. So people are like, genetic testing on embryos has become normal over the last 15 years. So almost every center in the country does genetic testing. It's just that the data that they're getting is much more limited. So they're just telling you about extra or missing chapters versus Orchid's kind of going much, much deeper and telling you for every single gene or sentence, or every single word or every single letter, has that been spell checked? Is it normal or abnormal? And basically, is it going to lead to severe disease? Either because unfortunately, some of these microduplications and deletions, they lead to even problems during pregnancy, right? So that some of these conditions mean that, that pregnancy is actually gonna proceed normally. So again, if you're, you've had multiple miscarriages, right? A ton of miscarriages, almost about 50% of miscarriages actually have a genetic reason why they occur.
Wow. Yeah.
So, you're gonna
Yes.
get this data hopefully on your embryos. Anyways, why wouldn't you want to get the maximum amount so that you can make the best decision, not only for pregnancy, but so that child doesn't go on to develop disease. Like we've unfortunately have folks who come to us who they've had to bury their first child before kindergarten because again, if you have one of these typos, in a really critical region, you might have a muscle wasting condition, or you might have condition where the brain doesn't form normally, and because they're so rare, there's such few people have each specific condition. Pharma doesn't actually go and, create cures or treatments because there's actually not a big enough market to actually sell a solution. So 95% of these diseases don't even have treatments, much less cures.
Wow. Yeah. I mean, for couples that have done IVF, they're doing these screenings anyway, and oftentimes they have plentiful embryo options, and you may as well choose the one that is the healthiest, or at the same time, make sure that it doesn't have any illnesses that are going to prevent it from living a life that is, healthy. And
yeah, no, I think it's kind of crazy. Like genetics is actually super, super unfair, right? If you think about it, like you and I won the genetic lottery, right? Like we got to, grow up and become adults. Like not everyone is that lucky. Like people, like my mom lost the genetic lottery, right? Like her life just got totally hijacked. And she lost her vision due to something that was completely random, right? And that happens. And she's actually one of the fortunate ones, right? That didn't happen to her until adulthood. That genetic lottery can, affect, not even making it to, month one of your life, right? Yeah. So it's actually like incredibly unfair. Like it's one of like the biggest, I think things that are unfair that are left in life, right? It's that there's like totally different starting lines, that people unfortunately start at. And it's kind of insane if you think about it, right? You're rolling the dice on the most important decision of your life, right? Like when you have kids the old fashioned way, you're literally rolling the dice on your child's genome. And I think it's actually, the most important parenting decision happens before birth, right? It's like probably the two most important parenting decisions are who your partner is going to be. And then the second is your child's genome, right? Because, people, over fixate on like preschools and like, organic food, but, none of those are as significant as, does your child develop pediatric cancer or not, right? Like it's crazy how important your genetics are, but, previously, there wasn't a way to actually, mitigate risk or to know about any of these things beforehand. And it's kind of crazy to be at this moment in history where, this information that previously you couldn't get access to until after, baby was born or until you're an adult, is kind of like, it's almost like this like fortune telling like capability that we have from, not just what Orchid is doing, but because, geneticists over the last 2 decades have cataloged all these different thousands of diseases.
Yeah. So what are some of the top diseases or conditions or predispositions towards these conditions that you look for?
Yeah, so I think a lot of people are really interested in genetic forms of autism. So there's things called like Rett syndrome, Fragile X. There's a lot of actually genetic forms of autism, and these are typically the most severe forms that are, folks that are, nonverbal and aren't able to live independently. I think that's like any type of neurodevelopmental disorder. I think people are really interested in. These are, unfortunately, infants that are born with seizures or epilepsy, intellectual disability, developmental delay. There's been these panels are actually provided to folks in the pediatric setting. So once a child has a clinical or behavioral diagnosis of autism or developmental delay, they'll get sequenced and they might find out if there's a genetic cause. It's just that Orchid is taking those genetic panels and moving them up to the embryo stage.
Yes.
So That's, I think I'd say one category that people are really interested in. Another category is birth defects. So there's things like, craniofacial anomalies. Heart defects is probably one of the biggest category of birth defects. There's skeletal anomalies. Sometimes you can see these on the first or second trimester scan. So we don't want you to be there, right? We want, that condition to hopefully be totally avoided, because a lot of those are, again, not compatible with life. And it's like a really, it's a really traumatic process to find out about those
later on
yeah.
Wow.
And then the other big category I would say is hereditary cancers, right? So there's pediatric cancers, so things like retinoblastoma, Li-Fraumeni syndrome, Fanconi anemia. And then there's a ton of genetic forms of adult onset cancers. So I think a lot of people have heard of BRCA, like breast and ovarian cancer. There's all sorts of other organs, systems that unfortunately can get cancer, like pancreatic cancer.
Super high risk for those types of cancers.
Yeah, exactly. Parents can kind of pick and choose what they want, what information they want. So they can choose, do they only want to know pediatric onset? Do they want to know adult onset too? Do they want to know about only those monogenic or like single gene typos? Do they want to know the genetic risk scores? Because the genetic risk scores, you're not taking your risk to zero, right? Unfortunately, you and I, we have probably the average or base rate for a bunch of different diseases, but the difference is, do you have a very significant genetic predisposition or not, right? So kind of when I was talking about that marble analogy, it's sort of like the way that you can think of disease is kind of like there's like a threshold effect, right? So let's say you have a bunch of marbles in a jar. If they're only halfway full with genetic risk, let's say we add another, 10 marbles of lifestyle risk, right? So I'm eating Twinkies all day, right? Like let's say I have half the jars full of genetic risk, and then I have, another half the jar full with, eating Twinkies all day. Now I've hit the threshold effect where the marbles are coming out of the jar, and now I've hit the disease, right? Versus what you're playing with genetic risk is how many marbles am I starting with?
Right.
Am I starting with 5% full, or am I starting 90% full? Right, where it's just one Twinkie is to take me over to, it's obviously dramatic, but basically you can think of it as like, what is your starting point? Are you starting, with that jar full, half full, a quarter full? The goal really with Orchid is that, we want every baby to win the genetic lottery. We want every baby to be genetically blessed. And, I think I think everyone we're surrounded by is unfortunately just genetically blessed beyond belief. Like they don't realize how many things could have gone wrong just for them to be born, right? Like your father has created trillions of sperm in his life. Your mother has started, at birth with not at birth, sorry. As an embryo, she started with 9 million eggs. By the time she was born, she only had 1 million eggs. Then by the time she hit puberty, she was probably at close to 400,000 eggs, right? Oh, okay. So there's this massive funnel to and it's super unlikely that you even exist at all. Like there's literally trillions of people who are not you that could have been born. Yeah. And then so think about how many men your mom met, right? Like thousands of men that she saw in her life, right? And she picked your dad. But then even once she picked your dad, there were trillions of other sperm and millions of other eggs that would not have been you, right? Yeah. And then that specific egg and that specific sperm had to combine, right? So we're now talking already about 1 in a trillion chance, even when you constrain to you and your mom and dad. But then, there's about a 30% chance of miscarriage, even after implantation, right? So when an egg and a sperm meet, not every egg and sperm that meets creates an embryo that actually implants onto your uterus, right? So that's actually an event, an implantation event. So that had to happen. And then the implantation event had to succeed. And then, you had a 30% chance of miscarriage even then. So it's insane how unlikely it is even to be born at all. And then to be born and to not have all of these, again, devastating diseases that, medicine is great. I really hope it continues to advance. But again, 95% of the time, there's not even a treatment. We've only cured, I think, a handful of diseases ever in medicine, right? We had, the smallpox vaccine, that was really, we've eradicated smallpox for the most part. We had the polio vaccine, which eradicated polio for the most part. There's still, a handful of cases, but with embryo screening, it feels like this is really, really exciting opportunity where it's not just, 1 or 2 diseases, but it's actually thousands of diseases at birth that your child just doesn't have to, worry about. Yeah.
Wow. I'm actually curious about the rates of successful implantation from Orchid babies
Yeah.
versus regular IVF testing babies versus, I guess, just natural. If you know those stats of like likelihood of actually implanting and causing a successful pregnancy and birth.
Yeah.
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 Because you were saying that the what was it, miscarriage risk is higher, or it's correlated with genetic issues, correct?
Yeah. So about 50% of miscarriages are genetic, but the thing that's really surprising that I think most people don't realize, people think that like you can just get pregnant tomorrow, right? Like I think most women spent, most of their reproductive years trying not to get pregnant. Like, all of the different, birth control options are like, maximum interest. But, you only have about a 10% chance of getting pregnant per month. Like there's only, 5 or 6 days where you can get pregnant. It's not like you can just get pregnant any day,
Yeah. Yeah.
So it's actually really, really impressive how good IVF has gotten, right? So over the last 40 years, like all the different techniques around, vitrification, embryo culture, media have gotten, I'd say a few percent better every year. So now you're kind of on par, honestly, with natural pregnancy rates, right? So you're, a healthy young couple has about, around a 10% chance of getting pregnant per month, right? So the way infertility is defined is that you've tried to get pregnant, you've been trying for a year and you weren't able to get pregnant. I mean, the definitions are shifting a little bit, but most.
I actually just want to have a quick story here. So a good friend of mine, she was considering freezing embryos with her partner in her early 30s.
Yeah.
And she pulled me aside. I was like in my mid 20s at the time. And she was like, Harper, definitely get your eggs frozen. I'm freaking out right now. I'm infertile. And I was like, what? And she was like, I went to the doctor to get my eggs frozen and I only have 4 follicles and I'm 31 or whatever. And they diagnosed her as infertile and she was devastated. It was such a horrible thing. They wouldn't even freeze her eggs because they're like, we're not going to get any.
Yeah.
And she was so sad. And then she was like, I'm just going to try naturally with my husband. Pregnant on the first month.
Amazing.
Like, this woman was not infertile, and so I just...
It only takes 1 egg to make a baby.
Totally. And so I just feel sad because I feel like some of these clinics I've heard just kind of scare these women into thinking like, oh, I don't have that many follicles. I'm going to be infertile or, I don't know. I froze my eggs actually a few years ago.
Oh, cool.
And I have more follicles now. I went and checked them again recently. I was considering doing it again.
Yeah.
And I've twice as many now that I had.
Wow.
4 years ago.
What's your health routine? Tell me.
Honestly, I think I'm just healthier now.
Yeah.
I exercise more. I eat healthy. Whereas back then I was like, I wasn't unhealthy. I've never been like, I've always eaten really clean.
Mm-hmm.
I think I'm just like, I was happier exercising. I think what I'm saying is because back then when I got checked, I was like, oh no, I've like half the normal rate for someone my age or something. And it made me so nervous. And I was like, oh, I'm going to be infertile. But for the women out there who are getting their follicles checked and potentially freaking out about it, it's less of an indicator of fertility than I think they try to tell you. I'm just going to throw that out there because it's caused a lot of stress for a lot of women I know, including myself. So.
Yeah. And I think that's other really good point is just like your diet and your lifestyle and your stress levels have a massive impact on your fertility.
Totally.
Right. So it's like, maybe if you didn't have a successful egg retrieval 1 month, like I wouldn't, give up on it. Like you can, make changes to your diet and exercise and hopefully improve your results.
Yeah.
And I do think like it honestly still was really great that your friend got checked because she might have waited longer and then, those 4 follicles could have turned into,
or it could have doubled into 10.
Yeah,
8 or 10,
exactly.
But it was, you never know.
But it was great that she had a baby then, right?
Yes,
like,
totally.
Like I feel like information is always just useful, right?
100%.
So yeah, I think that, a lot of, the folks that have, worked with Orchid have told us similar stories of like, they had no idea that, for whatever reason they had like premature ovarian failure or PCOS or endometriosis. All this stuff is really underdiagnosed until you actually go in and check. Totally. And just knowing earlier is just better than knowing later.
Exactly. It's good to go get checked.
Even once you have embryos, you still don't have a baby, right? Like you gotta bake that into a baby. That process still takes 9 months. So
it totally does. Wait, okay. So back to the question of implantation. I am curious about this. Do you happen to have those stats of like proper implantation with Orchid versus regular IVF? You said it's like 10% for regular IVF.
No. So sorry. So implantation, I think the national average for implantation is around 60%. So basically, like when you transfer an embryo, you have about a 60% chance of getting pregnant. But I don't like averages because it's a little bit like saying, what's the average price of a car? A Toyota Corolla and a Ferrari. Okay, well, the average price. So because it's very bimodal, right? If you think about like young, healthy people who are doing IVF, not for infertility, but maybe because they have a genetic disease, you know, I've seen centers reported as high as 80%, right? And then there's other people who are, you know, unfortunately 44, they have premature ovarian failure, you know, they have different issues with their eggs. For them, it's 0%.
Yeah.
You know?
Okay,
so
I see what you're saying. It's also in the person. It's not just the genetic factors. Yeah. I guess I was just curious.
There's uterine factors as well, right? So depending on, you know, your, you know, endometrium, there's a lot of, unfortunately, it's a complex, amazing, beautiful system, right? It's like really a miracle to have a baby. I think everyone acts like so casual about like babies, but it's like, no, this, like, think about it. I mean, I can show you the lab, I'll send you some videos, but it's like there's a single cell and it turns into a trillions of cells that are all perfect and magical, right? They all work together. Like it's a single cell that's somehow differentiated into a brain, a heart, a lung, and then just from nothing. I mean, they can show you a petri dish and then it's suddenly a baby. It's like totally insane. Like we totally don't understand how that works. And, you know, the fact that, you know, pregnancy itself is like the most insane marathon, right? Like literally you are at your maximum metabolic limit, like the same, you know, metabolic limit that like ultra marathoners or marathoners, you know, are measured to be at. That's what pregnant women are at every single day of their pregnancy. So it's like we have really pushed the limit. And also you think about like baby head size. It's like the maximum size that
possible that it can be.
That it can be to leave the body. Yeah. And like we still have such a high C-section rate, which obviously has, you know, multiple reasons that aren't just you know, baby head size. But it's like amazing that it even works at all. I mean, it's truly magic. It's truly magical.
Yeah. Yeah. Oh, I guess I want to pivot towards, not really pivoting, but you've said that your hope is for everyone to ultimately run this genetic testing on their embryos. So right now, as far as I understand, you are not testing on already implanted babies. So women who get pregnant naturally cannot do Orchid testing. It's only pre-implantation.
Yes, that's exactly. So you have to create embryos and do it via IVF. Yes. Because the challenge is, you know, if we were to offer this testing on, you know, 10 or 12 weeks, right, the only option at that point would be termination, which, you know, I think is really challenging from both, you know, an ethical standpoint and from like just a physical standpoint. So when you do it in the IVF setting, you're basically preserving your future fertility, right? Because a lot of people have, unfortunately, secondary or tertiary infertility, meaning they're able to conceive at home for their first baby, but then for their second or third, they have a lot of issues. So basically, you know, the way that I think society is moving is that I think people want to freeze their eggs and sperm at the youngest possible age and then create embryos so that they can have whatever size of family that they want. And that, again, they're also not, you know, rolling the dice on their child's health and their child's genome, right? I mean, if you know, have kids the old fashioned way, you're unfortunately going to be exposing them to, you know, 10 times the risk as if you were to scan their embryo's genome, right? Which sounds kind of crazy, right? Like, you know, it's funny because, you know, we're so used to doing that and not thinking about it as a risk. But, you know, there was a similar response to hospital birth versus home birth. People are like, oh, well, why are you over medicalizing, you know, pregnancy and having birth, you know, giving birth in the hospital? Well, it's like, well, you also have, you know, infant and maternal mortality rates because now when you're hemorrhaging or there's all these, you know, adverse events that happen during, you know, childbirth, there is, you know, the medical infrastructure there to be able to intervene. And of course, I'm not against like home birth or anything. Like if you're low risk, totally do it. And it's like your decision. It's your, you know, it's the experience that you want to have. But what I think is really cool now is that women have so many choices, right? You have this choice, home birth or hospital birth. You have this choice of, you know, epidural or no epidural, right? Yeah. And epidural was also introduced with a huge amount of taboo associated with it. Like there was actually a bunch of people who were against any type of pain relief during childbirth because of Eve, and they were saying that, well, you know, women are supposed to have a painful childbirth.
Oh yeah, because it was part of punishment, God's punishment.
Yeah, exactly.
Oh my gosh, yeah.
No, that actually put a huge damper on it, right? It was used in almost every other part of medicine, you know, for, you know, surgeries and, you know, dental procedures, but it was like, no, women aren't allowed it during childbirth.
This is part of their,
seriously.
Right.
And, you know, there was, you know, 2 different Supreme Court cases that were fought to give women access to the most basic type of birth control, condoms. Like doctors were put in jail in the US for giving women condoms, right?
It's radical at the beginning and then people over time.
Yeah. So I think it's like people just, I think eventually go toward where they have, more control and more choice and really the better health outcome for mom and for baby, right? I mean, I think if you've had multiple miscarriages, how emotionally and physically grueling those are, right? So if you can, more than, reduce massively the chance of a miscarriage by taking out the genetic, factors that cause miscarriage, right? That's just a pregnancy. Now you're talking about, you're going to be raising this child for, more than 18 years. Hopefully, you're going to be have a relationship for the rest of your life.
Yeah.
And I think for people who've seen how, their lives get totally hijacked by a health event, the last thing that they want is for that to affect their child. And now we know so much more about the impact of genetics. And of course, I don't, genetics isn't the say all end all, right? Like you should obviously still have a really healthy lifestyle and diet and all this stuff is like insanely important, but it's not an either or. You don't have to not have a healthy diet and lifestyle. It's just that, do you want to also have, your child be, essentially genetically blessed, be a super baby, right?
If you have this data and you're already doing some testing anyway. And also I think another thing I want to point out, so obviously, embryo creation and freezing and egg retrieval, they're expensive processes now. But over time, as the technology advances, it's going to become less and less expensive, more affordable for everyone, right? That's the goal, as you were saying, because you would like everyone to do it. And obviously, it's very expensive right now. It's like $10,000 for an egg retrieval, and then there's the embryo, and then whatever.
Yeah.
And then the testing itself, I'm sure Orchid has its own fees associated, but over time, as the tech advances, it'll become more accessible.
Yeah, because right now our healthcare dollars that like you think about all the, money that you put into, health insurance and just how we spend our healthcare dollars today, it's completely backward because these diseases are extremely expensive, right? The cost of these diseases over your life are, millions of dollars for some of these diseases, right? And if you can sidestep them entirely, I think if people were to choose how do they want to allocate their healthcare dollars, I think they wouldn't want them all in the last 2 weeks of their life on a ventilator. I think they would want them during these really big moments like, pregnancy, egg freezing, embryo creation. And there are other countries that support, reproductive technology more. There are countries where you get free IVF until you're 44. And I think that it's insane, honestly, that we live in a country where, wealthy people get to have babies via IVF because infertility is something that affects everyone, right?
Yeah.
Unfortunately, 15% of people are, have issues with infertility at some point in their life.
The ovaries, they age twice as fast as the rest of the body, correct? I think there's like the tissues. I remember there's someone who's doing research on this where the ovaries age twice as fast as normal organs do, which is crazy. And so,
wow, I didn't know that actually.
Yeah, this early retrieval of eggs and creation of embryos. I guess you wouldn't create the embryos because you don't know who you want to create embryos with when you're quite young. Maybe you do, but a lot of us don't. A lot of us don't. Would really help women help women have babies later in life because it's actually, I've heard, it's not the body that is the kind of limiting factor on the ability to have babies late. Like in fact, older women can carry children. It's just the egg. It's the egg quality that degrades so much that they're not able to get pregnant.
Yes, exactly. The egg quality degrades. There's actually, I think the oldest ever woman to have a baby via IVF, I think was like in her 70s actually.
Oh my gosh.
So yeah,
had she had a baby before? I'm
sure.
curious because I guess I just wonder like if the body, because the body shifts the first pregnancy, right? And then makes it easier for consecutive pregnancies. I wonder if she was pregnant before or if that was her first time. But I just think that's amazing, right?
Yeah, but you think about how powerful your uterus is. That like at 70 years old, someone can carry a baby.
Yeah, it's just the ovaries that age.
Yeah,
it's
and
crazy.
the eggs. Yeah, but here's the thing. People are always focusing on women when it comes to fertility, but there's I think not enough attention on sperm. So sperm actually degrades. The genetic material in sperm also degrades. It's not like somehow immune, these magic cells, right?
Yes.
So men who are over 40 have anywhere from 2 to 5 times the risk of a neurodevelopmental disorder in their children because there are higher rates of de novo or spontaneous mutations in their sperm, right? In the same way that, unfortunately, when you have, exposure to the sun, you have, more skin cancer. What is that, right? Like one of the mechanisms is understood to be DNA damage, right? So unfortunately, that machinery that's, protecting eggs and protecting sperm degrades over time, right? So older sperm and older eggs both carry genetic mutations. It's just that the eggs, it tends to be that the mutations are more catastrophic, actually. They sort of don't lead to baby. The damage that accumulates in eggs, it kind of results either in embryos that, don't even make it to day 5 or that are, have chromosomal abnormalities versus for sperm, what happens is it's these subtler genetic issues. There's smaller typos that happen, but they end up leading to neurodevelopmental disorders because more of those typos are introduced in older sperm than in younger sperm.
So Orchid is actually really helping for those cases, for the sperm cases.
Yeah, exactly.
Yeah.
So you can actually track, where did you, this is a little bit, controversial, but you can kind of see like, okay, did you inherit it from mom or dad? Who gave you, where did this issue come from? You can find out.
Where you're pointing the finger.
Yeah, exactly. But I mean, the reason why that's useful in the IVF setting is that sometimes what if it affects, all eggs or all sperm, then sometimes people consider egg donor or sperm donor in those situations. And then they're able to have a,
informed decision, more data.
Yeah.
I always say like more data is better. And if you're deciding on a decision, I've seen somewhere that they say if you're not sure, if it's not a solid yes, then the answer is no. But for me, it's just, if it's not a solid yes or solid no, then the answer is get more data. So yeah, I wish people,
and the thing that's like really wild about this is like people get so much information for like the most trivial decisions in their life, right?
Totally.
Like I've spent so much time researching like a lunch or dinner spot. I don't want to support my friends. I'm like, okay, we got to go someplace good, right? Yeah. And it's like, think about just how like, I mean, we've just rolled the dice on your child's genome. Like it's going to be with them for life. We don't have the editing technology to be able to change every single site, right? And they're going to have that for life. And it can either, be a healthy genetically blessed genome, or it can, unfortunately have a typo, right? Wow. So it is like I think, a weird mental shift for people that, hey, actually parenting starts pre-birth, right?
Yeah.
But it's kind of crazy to think about it, that like the most important parenting decisions could actually happen before birth rather than after birth, because they're kind of immutable changes, right? Who you choose as the partner is going to immutably be that child's mother and father, right? That's never going to change, right? And similarly, that genome, unless, we get ASI and we get really amazing editing technology, is also going to be, stuck with them for life.
Yeah. I mean, it's amazing the work that you're doing. And I guess Orchid's systems are just expanding as you get more and more data. Are these data sets expanding? And maybe perhaps you are also developing your own? I guess I don't know if you would be.
Yeah, exactly. So basically, more and more people keep getting sequenced, and these study sizes get larger and larger every single year. So there's actually these like academic geneticists that create consortia, and they create something called like standard operating procedures. So they basically review variants and new data as they come in every year. So there's these certain categories of variants that are called variants of uncertain significance, meaning it kind of is what it sounds like. It's uncertain significance. They don't know whether it's pathogenic, meaning disease causing, or benign, meaning it's fine. So those, as we get more data on those VUSs, or variants of uncertain significance, those sometimes get categorized as either pathogenic or benign. So basically, what's one thing that's really cool about Orchid is that you can come back in 5 years or 10 years, and then we can do a reanalysis on your embryo's data, and then if whatever the latest information is, then you're able to find out more or fewer risks on those embryos.
And so Orchid is available worldwide at various IVF centers.
Not quite worldwide. Orchid's available at, I think, over 60 IVF centers in the US. Oh. But yeah, people do come from around the world already to use Orchid. So we have been really fortunate to have folks coming in internationally because of a specific genetic anomaly that only Orchid can screen for. Because that's one of the most rewarding things, right? Is that people will come to us, and every other lab or company isn't able to help them, but we are able to help them.
Yeah.
And sometimes those families come, from overseas, and hopefully, it won't be such a trek for them, and we'll be able to, have some more satellite offices pretty soon. But for now, US is a lot of folks, so we're just working our butts off to, service everyone here first.
Yeah. And some people I hear, I got a Prenuvo scan done, cool. And I got some people who are very upset saying, this is an expensive procedure. It's $2,000+. Why would you promote this? And I think what I would say to those people, and obviously Orchid is expensive as well at the moment, but what I would say to those people is like, the people who are the pioneers in using this technology are the ones who enable it to become more affordable for people later on. Like all of these scans and kind of scientific systems that we use now for diagnosis, they were all very expensive at some point before in the past. Yeah. And then over time they get better. And as people are using them, they become more and more and more affordable. So just because it's expensive right now doesn't mean it should be like poo-pooed, by everyone, because it's ultimately the goal is to reduce the price and make it more affordable for everyone. And that naturally happens in like in an economy where more people are using it.
No, yeah, I think it's really weird, honestly, the kind of stigma there is for these early adopters. I think it should be the opposite, actually, because they're actually being really brave, and they're being the first users of this technology. I mean, I'm extremely grateful to all of Orchid's earliest customers, because it's the most sensitive, most like high value cells you're probably ever going to create, and they trusted us with them. And obviously, part of the reason why I tested my embryos in our lab first was for that exact reason, because I was kind of like I built this company, this product, because I want to use it. And,
So you were the first person to use it?
I was the very first. Yes.
God,
I was the very first
that.
samples. And I just like it because, I wouldn't want to like recommend or like talk about anything, to anyone else. I wouldn't be able to do that if it wasn't,
right,
if I hadn't done with my embryos first. And I think that's the other thing that I find kind of really fun about this journey too, is like, if, whatever, the world rejects super babies, at least I got my super baby out of it. So,
I love that.
so I'm kind of like, it's the way that this all happened is literally just like me showing my embryo report to all my friends. And then I guess they all just got really excited about it, and they told all their friends, and like,
now I heard about it through a friend of mine. That's so funny. But so how many people have used Orchid now? And I guess honestly, we need to kind of wrap up. I'm so sad. I could talk to you for hours.
No.
But unfortunately, I have to go. But how many people have used Orchid now? And because I know it's still relatively early stages. I talked to you like a year ago about it.
Oh, wow. That's awesome.
And I think it was early on. But yeah, so how is it looking now?
Yeah, so we've tested, thousands of embryos now. So I don't know the exact number of babies, but like my entire inbox is just full of baby photos, which is like the coolest inbox to have. Honestly, it's like so cute to just be seeing. Yeah, I mean, like they've been calling them Orchid super babies now. So I'm like, that's cute.
Yeah. Oh, they are.
And I've been holding them, and
babies, holding the babies.
I've been holding the babies. Well, because I mean, so at the very beginning, I knew like every single Orchid user, and nowadays, unfortunately, I don't know everyone.
Yeah.
But some people still keep pinging me, and then some of them are like based in the Bay Area, and like I get to go meet them, and it's just like the coolest possible thing. I don't know. Sometimes I'm just like, it's very surreal. I'm like, is this really my job? I get to just like hold babies that that their parents are saying wouldn't have existed without our technology, and it's just like I don't know. It's just like, yeah, it's really fun to be able to. Like, I don't know, this moment in people's lives is just a really fun moment to be a part of, because it's a moment of like so much joy and excitement. It's like a new beginning, and it's just like maximizing the chance that not only that pregnancy, but that baby's whole life is going to be awesome. Like I don't know, I love getting to be a part of that moment in people's lives.
Yeah, and that couples who are older who are trying to have kids are more likely to be able to have healthy babies and things like this. I mean, it's beautiful what you're doing.
No, thanks so much. I mean, I love what you're doing too. I mean, I feel like there's, I don't know, I feel like AI is like super inaccessible to a lot of people. I feel like it's like something that only people who have like PhDs or something can understand. And I think it's so cool that you're making it so digestible and easy for so many people to understand, because it's going to be such a huge part of our future. I mean, it's obviously such a massive part of our present right now. But I just love, just like how simply you break down these concepts so that, people can really, feel like they understand like how all this stuff is built, because it is, the concepts are actually really simple, but it just takes, it's stuck in so much jargon. So it really takes someone special like you, who's like been a practitioner and been to all the classes to really be able to break it down that way. And I love it. I love it. It's really cool to see your stuff come up in my feed.
Oh, thank you, Noor. I'm so glad that you are here today. Thank you for everything that you're doing. I'm so excited about Orchid. I'm excited to keep following it and potentially using it someday. But yeah, thank you.
Thank you so much for having me. This is awesome. So fun.
Thank you for watching. I hope you enjoyed our conversation as much as I did. Let us know in the comments what you're thinking, if you have any questions, and I hope you'll become a subscriber and join our community. So until next time, this is HCAI, Harper Carroll AI, and I'll see you soon. Bye.